Hello, everyone. Welcome to the GEMS podcast. I'm so glad you all are here.
Today, we are so lucky to be joined by Dr. Lataisia Jones, a neuroscientist. Lataisia, we are so happy to have you. Could you tell us a little bit about yourself?
Likewise. Well, I'm excited as well to be here. So a little bit about myself. I have been a scientist for a little over 13 years. I originally started let me go back some more. I was born in Virginia to a military family, moved around to a lot of different places, finally settled back into Virginia, and I originally went to Virginia State University. And originally when I went to Virginia State University, I wanted to pursue pre-med. And I saw a flyer one day that was advertising a lab experience where it was inside of an internship at the College of William and Mary in Williamsburg, Virginia. So I decided to hopefully sign up. And after a few months of just being persistent and asking for the opportunity, I finally gained it and just started my research experience in the first lab I've ever been in. And I was working with C. elegans, which are microscopic worms, and looking at the molecular biology behind the research of my particular project. And then my research interest kept blossoming. So I continued to pursue other research experiments. I next went into a diabetes research project at Virginia State University, and that eventually became my master's thesis. And then I went on to my PhD, where I earned my PhD as the first African American to graduate from my program in the Department of Biomedical Sciences at Florida State University. So that's just a little bit about myself across the spectrum of everything.
Wow, okay. So is there an experience or person that drew you into this field before you started pursuing higher learning, or did you always know that you wanted to pursue STEM?
I actually did not know that I wanted to pursue STEM. So a quick little funny story. When I was in middle school, I was on a basketball team, and my family, especially on my dad's side, they're all like sports fanatics. So I was raised around basketball and football mostly. So it wasn't a surprise that I wanted to pursue something in basketball or whatever else. And my mom actually was talking to my teacher, my science teacher, and my science teacher complained and said, "Your daughter's talking too much in class, and that is the reason why her grades are failing." I wasn't failing, I just had like a C or D. But in a military family, a C is awful, right? In the military
That's right.
family.
Mm-hmm.
So, and because I was talking in class, it made it worse. So my mom threatened to take me off the basketball team if I continued to talk so much in class. And eventually that's exactly what happened. She removed me from the basketball team. So I had to focus more in class. I had to do better because I was thinking maybe this will allow me to go back on to the basketball team if I improve in my behavior within this class. And it actually turned out to provide me with a better experience because now I was fully thrusted into understanding and investing myself into a science. So I started becoming more interested and eventually joined the debate team. And the funny thing I tell people a lot of times, my first question that I ever got right on the debate team was about chlorophyll, something that I would have never known if I never took the science class and took it seriously. So, that's basically how I knew science was of interest to me. I still didn't completely commit to it until my college years, but I had that internship experience. But that would be the first time that something sparked and said, yeah, science could be a go for me.
Wow. Yeah, I'm so glad that you traded your hoops for a lab experience.
Right.
Yeah, no, that's great. So after your internship experience, you took a trip to Ghana, right?
Yes. So during my master's years, I took a trip to Ghana. So my advisor that I had during my whole majority of my undergrad and my master's years, he was also my research advisor. And I was also an RA, and I became a GA. So that's the person that oversees the residency dorms. And that meant that I was never leaving to go home for summer breaks or spring breaks or anything else because I was constantly working in the lab or working within the dormitories. And my advisor came to me. He said, hey, would you like to take some time off? And I never heard that before. I never even thought of that as an option. So when he brought it to me, I was like, actually, that sounds like a good idea. I just wouldn't know where to go. So he advised me to go out of the country, and he had this opportunity for me to go to Europe. But I had a lot of interest in figuring out where my family lineage was traced to. And in particular, my mom's side is derived from Ghana. So I wanted to learn a little bit more about Ghana, and if any opportunity came up for that, I asked him to please let me know. And lo and behold, another school was going to Ghana for a summer, and I applied for a scholarship, and it allowed me to go there without having to pay for room and board. All I had to pay for was my flight, and any money that I brought with me was to be spent on whatever things that I just wanted to bring back home from the trip. But during the trip, I had an opportunity to teach at two schools. I was teaching science, math, and English. And that was a very pivotal point in time for me, because I was able to see the excitement in the students when they were meeting an African-American scientist that was a woman, as well as I was able to utilize some of the things that I taught other kids in my community at home science through. So I was making science fun for these students, and because of that, it showed me that I wanted to continue doing that as time progressed. So it essentially allowed me to say I wanted to pursue a PhD from that day on. So I immediately, when I went home after the trip, I started applying for PhD programs, and that's how I ended up at Florida State University.
Wow. So what was the classroom setting like in Ghana? Like, how did you teach these students English? Were you communicating through a translator, or how did that whole situation work?
Oh no, so they already had the foundation, so it was very
okay.
I was only coming in like after they already had years of training, right?
Mm-hmm.
And Ghana in particular, one of the major languages is English as well, because it's so business oriented, and a lot of tourists come through there, that English has to be something that's integrated into the culture, because you don't make a lot of money if you don't cross over into that spectrum. So it wasn't as if I was going into a classroom where students didn't speak English at all. They already had the foundation. So I was coming in to basically help with them matriculating to the next level in their English vocab. But as far as science, I think the thing that was very tricky for them was weather. A lot of them had never seen snow before. So I literally went into my phone, brought out pictures of me in the snow when I was living, in whatever places that I lived in that snow, Virginia, New Jersey, wherever else. And I would talk to them about the experience of, like, my dad having to shovel us out of the house because the snow was so high. And it'll help them to actually imagine this way of weather, this cold, extreme type cold situation where all this snow builds up can actually have you locked in your house or blocked into a driveway, out of a driveway, in a driveway, whatever else. So it helped them out with understanding things that they had not seen as much because of being in a different country.
Wow. Yeah, down in Columbus, Georgia, we do not get any snow, so I definitely relate to that. So on your journey to gaining your PhD and afterwards, have you seen an apparent gap between male and female roles or anything like that?
Absolutely. So there's always a gap in females and males, right? Even before I went to college, I always blamed the fact that I wasn't as interested in the things that I'm interested in now, because I didn't see enough women and women of color, especially that were already in the lab setting, had a PhD or whatever else. And I often say that when I was younger, the only image I had of a scientist was Bill Nye the Science Guy. So in my imagination, you kind of had to look and fit his role in order to be in that scientific, background, scientific role. So that was before I even went to college. So definitely in my PhD years, it persisted. It just became a lot more extreme, because now I was going to national conferences, international conferences, and I was still seeing very few women compared to male, and definitely a lot less Black individuals or African Americans in these arenas. So it was oftentimes that I would go to, so my project was neuroscience based. So I would go to the Society for Neuroscience conference, which is a huge conference that has about 10,000 people there. And I could spot out which individuals were African American or Black in all of these people. And then when it came to women versus men, of course you could see when you go into conference rooms, meetings, or whatever else, the majority of the rooms had more men in there than women. So it has always been like that as far as the proportions. It's very scarce as far as being a woman in these fields or being a woman of color or just being of color in these fields as well. So minorities, the diversity in those arenas is very low.
Yes, and I wanted to highlight, just to make sure that everyone heard, but you were the first African American to receive a PhD from the Department of Biomedical Sciences at Florida State University back in 2017. So
Yes,
so you're doing your part by diversifying the perception of scientists in general for people of color and women, and I think that that's incredible.
Thank you. Thank you so much.
Okay, so could you tell us a little bit about your day-to-day operations and what that looks like?
Yeah, so as a neuroscientist, you're constantly reading other people's publications to see what the newer information is regarding the project or the topic of your interest, because you're looking for ways to answer the questions regarding your experiment or your experimental model or whatever disease you're looking into and trying to understand it in a better way using novel techniques. So that's always the number one thing, is just keeping up to date with better techniques, with the information that's already known and so on, so you can expand on your own experiment. And then using that information, you build and design your experiment to answer the questions that you have specifically for whatever you're looking into. So for me, for my PhD, I was looking at Levodopa responsive dystonia. And for those that do not know what this is a debilitating movement disorder that basically forces the limbs of the person into these awkward positions that cause pain onto the body. But the individual that's inflicted by this disease cannot untwist themselves. They don't have the control over their limbs in this situation. And the unfortunate thing is this disease has a child onset, which means it starts in childhood ages. So imagine being a child and not being able to play sports or ride a bike or even sleep comfortably at night because your limbs are completely twisting in different positions in these painful positions over and over without your ability to stop it. That was the disease that I was looking into. So in the process of my research, I was looking to find new information regarding this disease, but also on a finer level, I was using molecular techniques to see on a more in a more detailed fashion what's going on within the genes that I was looking at in particular. And that gene, I found out later on, had a mutation in it that was creating the wrong protein. And as a result, the dopamine, which is the neurotransmitter in your brain that produces what you need in order for you to control your movement, it was not working. It was not enough of being produced in these kids, which means that their controllability of those muscles or those twisting of the limbs and so on was faulty. So I created an antibody specific to that mutation in that gene that allowed us to specifically look at the human cells, the human tissue samples or whatever else to detect that mutation in the gene and help the families that were impacted by that particular disease to know early diagnosis of that particular disease. So that came about with using research and that other people did, understanding the stuff that's already out there, but also using new practices to understand what's going on in my own research.
So that antibody is used in genetic testing in order to catch the disorder earlier rather than later, right?
Right. So, and then the unfortunate thing is, it's one of those diseases that's very rare. So it's not a lot of people that's impacted by this, but the best thing about this is the families that I did help now have something to go by. Now when they continue to grow in their family tree, they could use that as a diagnostic marker for that disease, for their future kids and so on.
Right, and to my understanding, it's harder and more difficult to diagnose because it's similar to other movement disorders, and
Yep.
the symptoms vary. So your genetic marker, your antibody, is helpful to those families. Not only those families, but people who don't even know that they have it. Right. So
Right.
their parents don't have to have the gene, do they? Is it a recessive or dominant? Could you explain a little bit about that?
Right. So that's the thing about dystonias. They're all different. So with this particular one, if you have that mutation in that gene, you have that disease.
Mm-hmm.
So that's what this antibody allows you to do. It allows you to remove any other possibilities and say, okay, we detected this. There is definitely there. And the big thing about this antibody is not only is this protein being created, but the antibody is specific to that protein, that peptide, that protein that's being created, that mutant that's being created because of this faultiness in the gene. So if you see that in there and you see an increase of that particular protein in the system or in the cells or whatever else, it is very, very, very true to say, well, this person has the disease. So it allows, yes, dystonias are very common in a lot of ways. They're very different in a lot of ways. And yes, movement disorders are all over the place as far as commonality as well. So allowing a antibody to come in and kind of decipher or remove any of disbelief is probably the best way to go in that particular situation because of how a lot of times it is falsely diagnosed as other things. So yes, that was a good way of narrowing down, does this person have levodopa responsive dystonia, and in particular, this form of that dystonia that produces this particular protein.
And so when they used your genetic marker, that antibody, to find this diagnosis, what were the available treatment options for those patients?
Right. So my project ended before we were able to get to that part. I was just able to show that in human samples that the protein can be detected with the antibody. And then my part of the process finished there, basically. A lot of issues that goes on with these particular diseases is the fact that they're so rare and there's so few people that are affected by them that funding isn't as frequent for these particular disorders versus like Parkinson's or something that's more popular, more people have it than levodopa responsive dystonia. So the grants are more, they're more, people that are granting money for these things are more likely to give money towards that. And anybody and everybody knows research needs funding in order for it to continue. So to the least, what we were allowed to do was allow this particular family that provided all their family samples for, 3 lineages on their family tree. We allowed them to have some peace of mind. Now they have an antibody to say, hey, specifically, this is what's going on with my family. They have research that says, hey, specifically, this is what's going on in our genealogy, and then so on. But on a wider scale, I was able to provide research with guidelines or like a blueprint as far as understanding that this mutation was created because of some things on a molecular level that a lot of people don't even believe can happen. If you allow me to go a little deeper, I'll try to explain it at my best. But
go for it, please.
in any gene, you have an open reading frame at the beginning, which is transcribed and translated in order to create, the synthesis product at the end. On this particular gene, the mutation is created because of a different open reading frame that is created is a single nucleotide polymorphism, which means one nucleotide in that sequence changes, right? And it's different from the regular genes or normal genes. And because of that normal gene being switched to now a different nucleotide, it creates another open reading frame. So both of these open reading frames are competing with each other, and because one is more upstream than the other, essentially the one that's being used the most is the one that creates that mutant peptide. So because of that, you have that abnormality in the production of the the normal peptide that creates dopamine, and you have more of the protein that's not supposed to be there that we call the mutant peptide or mutant protein. So this is now a blueprint being provided to the research scientific community, and now they're using that research and saying, okay, this person showed us that if you have 2 open reading frames or more, it could create an issue within the gene and what the gene is supposed to synthesize at the end in order for the body to properly function. And in this particular situation, it was a lack of dopamine, but maybe in another disease, it creates a lack or increase of something else. And then now we need to look at that more specifically because she's showing proof that this could occur. So on a greater scale and on a bigger picture, that is the thing that created more of a contribution towards scientific community and the progression of society medically than the fact that I was able to make the antibody, mostly because more people will be affected by what I did in my research versus the antibody that was more specific to a family. You know what I'm saying?
Yes, absolutely. And so levodopa responsive dystonia is caused by the lack of dopamine, not the excess of the other protein that was being created. Is that a fair assumption?
We don't know.
We don't
know.
Okay.
We don't know what the effects are of having that protein created in the system. We didn't see any issues. I looked for cell abnormality. I looked for maybe misfolding or anything else. It did show that some slight misfolding. It didn't show much cell abnormality or anything. There was no increase in cell death or anything. But it would take a little bit more research to understand that better and something a little bit closer to the human samples of the actual patients, just to get a more better means of comparing, I guess I'll say. So we're not sure if the mutant peptide aggregates or mutant peptide production is the big issue. We do know for sure, though, because the mutant peptide is being created instead of the normal protein, that is causing the lack of dopamine or a decrease in dopamine, which is the overall biggest issue in that situation.
I understand. So, to my knowledge, after you received your PhD, you started focusing still in the Department of Children, Neurological Disorders, things like that, specifically autism. Could you just tell me a little bit about any of the research you did with that?
Absolutely. So with autism, I was looking at the corpus callosum, which is the middle part of the brain that connects both of the hemispheres, and it's responsible for that interhemispheric communication between both of the hemispheres. So without that connection or that bridge, your left side of your brain wouldn't be able to talk to the right side of the brain. And essentially, I was looking at what's going on in the postnatal development of a child, but I was using a mouse model in particular, and I was looking at proteins that are already associated with autism. So in particular, I was looking at the plexins, and plexins are known to be very ambiguous as far as what they do and how many other plexins they group up with in order to do what they do. So in short, I was looking at specific plexins to see what was going on with the corpus callosum, and specifically looking at neurons and their transmission of information into the corpus callosum and whether or not the corpus callosum size was faulty as a result of faultiness in the plexins. So just seeing if differences in the protein that's expressed would would kind of counteract with the ability of the corpus callosum to work and function properly.
Wow. Okay. And you said now, after your new job in March, you're starting to work on reading research papers and proofing those and things like that. Could you just tell us a little bit about how that whole process works?
Yeah, so this is my first time actually leaving the bench, the scientific bench, in order to pursue other things in science. So I am now an ethics fellow for the American Society for Microbiology. And as an ethics fellow, I work with publications to look at papers that researchers are basically putting out into the community and publishing in these journals to show the progression of their research on a particular research topic. And my main job is to make sure that ethically, whatever is being published by ASM, or the American Society for Microbiology, is sound and good data, as well as has no manipulation within the data as well as the text and so on. So I'm looking for plagiarism. I'm looking for false data, duplicate data, data that's been spliced and so on, to make sure that whatever we have published is something that society could really use to look into the progression of the medical field, or just in anything. Right now, the biggest thing, of course, is COVID-19 research. So ASM has expedited COVID-19 research to make sure society can get it in a more timely fashion than usual. So this means that on my desk every morning, if there's something related to COVID-19, that is a priority. And we try to work very well and still in the same esteem, but make sure that this information is being provided to the society, so that way the progression of figuring out vaccinations and anything related to the loading, docking, or just the structure of this virus can be progressively worked on in a faster fashion in order for us to figure out the best way to keep humans alive when it comes to this virus. So that's how important this particular arena is, and I never knew this beforehand because I was so stuck to working in the lab, and I thought, now I'm a scientist and I have to be in a lab all the time, but I'm using my scientific experience to now improve the information that's going into society through publication. So that's what my position entails now.
And so in that position, if it were a non-pandemic world, I know that you took it in March, and that's when it was coming about in the States. If it weren't in that kind of situation, would you specialize in a specific area? Would you have a specialization in, neurological disorders or children? Or is it just you read anything and everything?
Yeah, so in this position, I'm reading anything and everything. So sometimes I get papers in reference to viruses. Next time it'll be a specific bacteria. I also have, we have antibody papers. So we're all over the place. And then as far as clinical research and so on, of course, like that'll be across the board as well, whether it's adults, kids, whatever else. So yeah, now at this point, I'm reading over anything and everything. I know in my past research, I was specifically focusing on kids and neurological disorders and anything in relation to those particular topics, but now working with the American Society for Microbiology, I'm a lot broader as far as what I work on, which is it's good because it's allowing me to learn in so many other ways and fashions about other topics that I probably wouldn't have learned about while being a researcher. Because a lot of times you get so focused on your project that things that are going on outside your project you don't get as much interaction with, or you don't learn as much about because it doesn't completely relate to what you're doing. So it's pretty cool to be able to say, hey, I'm still reading papers, still keeping up to date, and I'm even still publishing based on what I'm doing within my position as well as my community endeavors when it comes to diversifying STEM and making initiatives that create that diversity in STEM and advocacy for diversity in STEM. So I'm still doing the same thing that I would do if I was in the lab. The only difference is I'm no longer in the lab.
So when you read over these papers, you're looking for, mistakes or errors or dilemmas that might make it plagiarized, things like that. Do you have to replicate these experiments on a small scale? Is that a part of your duty or your ASM as a whole?
Right. No. So actually there's software that allows you to look into these images that's presented on the submitted manuscripts to look for any duplication of images, which means images that have been used over and over to prove different points or to provide different conclusions and results. as well as slicing of images, which means something was cut out, an image or data was cut out of something else, but wasn't properly cited or shown that it was originally a part of something else. So there's software that allows us to look into these things. We actually have image specialists who take their day to analyze these images and make sure that anything that comes up that looks like suspect data is sent to me, myself, and then I take it from there. I look at it again to see specifically what the issue is, go more in depth to understand what the project and experiment was about, to understand what the data was that was presented within the figures. And then if it does look like something that needs some type of clarification or some type of modification, I contact the authors. And usually it is just a mistake that they made, because according to ASN's guidelines, if you do certain things such as splicing an image, you have to show that the image was spliced from something, because you have to think about it as a reader. If I'm reading something and I see this image that looks like it came out of something else, I'm not going to believe the data because I don't know how it was composed, or it may give me a false impression of something else if it was done to be manipulative. So as an ethics fellow and in my role, I go talk to the authors and I say, hey, we saw this potential issue. Let me help you fix it. And they provide me with additional information. And then if it isn't something that was done out of malice or out of deceit, we go from there and fixing it. And then if it is something that is done to manipulate data or so on, we not only let the institution that they're working with know, but we also make sure that that information is not put out into society, to make sure that what we, like I said, what we produce and what we put out there in our journals is something that readers can use to understand more about diseases, disorders, antibodies, clinical trials, whatever else, without feeling like they're being manipulated or without looking at false data.
What you do is really making an impact on us readers who, get to receive that information and knowing that it's credible. And you mentioned that you work with educational things to prompt the discussion or diversify the representation of STEM. So if you would want to just talk about the things that you do, I know that you work with the If Then, you're an ambassador for that, and just if you want to talk about a few of those things so people know and they can be exposed to that and look into it if you want to.
Absolutely. So I am a AAAS If Then ambassador, which is a Lyda Hill philanthropy initiative in order to provide girls in middle school and high school with motivation to pursue STEM careers. Lyda Hill basically came up with this initiative to say, hey, I'm looking for women in STEM. They don't have to just be in a career. They could also be in academic programs, but they have to be amazing mentors and willing and able to do what is necessary to get young girls interested in STEM careers. So there was a lot of applicants, and out of all of the applicants, the committee, the councils, which consisted of middle and high school girls, as well as the individuals that are working on the staff, picked 125 women out of the nation in order to serve as ambassadors in this program. And we are the first cohort of women in STEM for this program. So what we do is, using that platform provided, we use media, social media, training, creation of events, grant opportunities to fund more events, to basically say, hey, we have this platform, we're going to use it to get more girls interested in STEM. So out of these opportunities, I've been able to star on a show called Mission Unstoppable, where it's on regular TV, and it stars women in STEM, and it comes on every Saturday. And Mission Unstoppable basically allows girls to see these women in their actual careers. So during my episode, I was aired inside of my lab while I was doing my postdoctoral research, and I was showing these girls or whoever was tuning in what it meant to be a neuroscientist. So they not only got to see me within the lab and, doing DNA extraction and stuff, but they also had a chance to see me at the microscope, and they saw a little bit of my data as well. So if anybody's interested in watching that, that is Mission Unstoppable. If I'm not mistaken, it comes on every Saturday, and it's on CBS. And then a few other things that I've done. I've done an interview on the news in the local area that I live in D.C., and just to talk about advocacy for girls to pursue STEM careers. I've done radio station news interviews. I've done a few things for Children's National Hospital, where I've done interviews, podcasts, and like commercial series. And then now I am in the creation phase. So another thing that's coming out next year, they provided full-figure statues of all the women that are ambassadors in the program. And this is to change the demographic, because the majority of the statues in the United States right now are currently all men. And when you look at the numbers, I forgot what the statistics were, but I think it was only a handful that were women. And if you could think about how many statues you've ever seen, that is very few compared to all the statues put together in the whole country. When it's safe to go back outside and go back around people, they will have all of our statues, all of our full-body statues at North Dallas Park. And all of the statues is going to be in this wide field so girls can walk through and see all these women in STEM and what they do to contribute to STEM. So that's like that grand surprise yet reward for being a mentor. So now that I am in the creation phase, and I kind of been creating more and more since October, I've created a few things on my own. So in October, I started Young Scientist Wednesdays at Children's National Hospital, where I basically bring the patients that are on chemo or whatever else, that's staying there for long periods of time, together every Wednesday so that they can learn something that's scientific through a fun activity. So we've done anything from exploring artificial augmented reality to look inside of a human brain. We've done ceramic or pottery to understand the science behind that, like centrifugation and so on. We've made snowmen inside the building during a 90-degree day just to show how what mixtures and solids and liquids, understanding those things a little bit better. We've even created little explosions or little bubble over messes, looking at acid-base reactions to make rainbow volcanoes, or even the elephant's toothpaste experiment to look at exothermic reactions and understanding what a catalyst is. So these kids don't only learn about the science behind the experiment, but they get to actually do it, which is more fun than just watching in a lecture. So that was my first program that I started while in the program and while, just creating stuff on my own. And when the pandemic started, I said, well, I can no longer be around the kids as much because they shut down the hospital in a lot of different ways to keep things safe. So I started a YouTube show called Kitchen Science, where I'm teaching kids science from the kitchen. So you're utilizing things from the house to understand more about STEM. And all the experiments that I explained from Young Scientist Wednesdays you can make those at the house too. So I basically have those same things, but on a YouTube channel. That way you don't have to be around me. I don't have to be around you, but you can still learn just by viewing into my YouTube. I also do live experiments, live shows through Zoom or whatever virtual platform. I've been doing that with a few different groups, a few girl mentoring organizations such as Girls to Divas and Crowned Academy. Girls to Divas is in Florida, Crowned Academy is in Virginia. I've also worked with the Detroit Boys and Girls Club and Hampton University. So these people are in different places everywhere. So that I was able to show them science through these fun experiments. They did the experiment with me. We were able to see each other and all of us still be safe from our homes. And then the more recent thing that's really been taking everything by storm is my Stemming While Black initiative, where I have used the virtual platform to encourage current Blacks in STEM or minorities in STEM or anybody that's just facing challenges from just being the only one or being one of few in whatever field they are in or whatever academic program they're in, with encouragement and with advice from current Blacks in STEM. So I have a panel of about 20 individuals who are all African American or Black in STEM careers and academic programs, and they provide advice based on whatever the topic is for the particular panel discussion. Anybody that registers gets to supply their questions through the registration portal. And the first episode, we talked about a lot of the questions. The second episode, we focused on the graduate student. And the third episode, we split it up into two parts where we talked about the challenges of being a Black man in STEM and the challenge of being a Black woman in STEM. And that allowed us to more specifically hone in on the issues of trying to increase diversity in STEM with all these challenges that are present. So from there, we are still continuing to evolve in all of these spaces, whether it's Kitchen Science, Young Scientist Wednesdays, the virtual experiments, me mentoring one-on-one, as well as Stemming While Black. And I created my stemming consultant company called Hey Dr. Tay, LLC. And from all of these things, of course, you can find all of these things on heydrtay.com. I know it's a lot.
You keep yourself busy.
I do. Because it really is motivating to know that, well, it's discouraging to know that I didn't see anybody that looked like the grown-up version of me when I was in middle school. And that's why I thought basketball was the thing to do, right? And then it took for me to act poorly within my science class in order for me to find a way to start focusing on it. And then I waited all the way until college, and that's when I found my first lab experience was the first time that I met a woman that owned her own lab. And then my freshman to sophomore year, that was the first time me meeting an African-American woman that had a PhD. What if I never had went to college? I was raised in a military family, so what if I decided to go to the military instead? I would have possibly never been the first African-American to graduate from Florida State University in the Department of Biomedical Sciences. So that would have waited until this year when the second Black person in STEM graduated from that program. So we would have been in 2020 versus 2017 with history that should have been made. So I always think about those things. And now when I work towards all these initiatives, I say to myself, well, this is what the kids need. This is what the youth need. This is what current graduate students need. They need exposure to diversity, need exposure to representation within these fields. And it needs to be a normacy. At the young level, you need to see that there are people come in different shapes, sizes, colors, genders, whatever else within these fields. So it should be normal to see that. Therefore, when you get there, it is normal to say, okay, I fit here because I've seen this before. And then I provide that opportunity now by showing you on Stemming While Black panels, Black faces in STEM. I provide that opportunity through a collaborator, and I have this program called Couch Conversations Between Kids and STEMioneers, where we use Zoom to show kids these STEM careers and give them an activity that's fun for them to do. So they get exposed to those people, too, that are grown up that they may or may not look like later on. So all this exposure and all of this encouragement and networking and advice and just going over the experiences these individuals go through help other people stay determined and motivated to do the same and understand what it really should look like before they get there.
Well, your inspirational hard work does not go unnoticed. I mean, it's really making a difference. That is incredible.
Thank you. I appreciate that.
Well, as we come to a close, is there any piece of advice that you would give girls for pursuing a career in STEM?
Absolutely. So the biggest piece of advice that I will always say to girls pursuing careers in STEM is do not give up just because it looks difficult or it feels difficult or it is difficult. There were many times where I wanted to give up. It's never going to be easy. And a lot of people don't tell you that after they've already, received their greatest accolades. They talk more about the things they've earned and what they've gotten themselves up to, but less people talk about the fact that it was hard at certain periods of time. And it's not meant for everyone, so that is why it's difficult. But if you don't give up and if you stay determined, you will find your way to whatever you're striving to be at. So goal-wise, set the goal, execute the plan after you create the plan, and then just keep going until you have received everything that you have set out to get. Don't give up.
Wow. Well, Lataisia, thank you so much for joining us and paving the way for underrepresented groups in the world of STEM. What you do is amazing. And on behalf of all GEMS, thank you so much for sharing your experience and wisdom with us. We really do appreciate it.
Thank you so much. And if anybody ever needs me in the future, I am Hey Dr. Tay on everything. Facebook, Instagram, Twitter, and my website is heydrtay.com. That's HEYDRTAY.com.
Perfect. Thank you so much. And I'll make sure to include that in the description so people can just click on it and all that kind of stuff.
I appreciate it. Thank you so much.
Thank you all for listening. If you have any questions or comments, you can contact us through Instagram, Twitter, Facebook, @ColgaGems. You can also reach us through email. Our email is colgagems@gmail.com. That is COLGAGEMS. And I'll see y'all next time.